PP Simmons family. Well, here it is. The very first PP Simmons broadcast show. Well, it's not a broadcast in the sense that it's being transmitted over the air, but it is a broadcast in the sense that it's being broadly distributed across our YouTube channels and Facebook and Twitter and the blog, and so welcome. I mean, isn't it amazing how far this thing called PP Simmons has come over the years? I mean, it all started with a few videos on YouTube in October 2008, and now it's spread all over the world. We have the YouTube channel and the blog and Facebook and Twitter and millions of views, and we're incorporated with Freedom Friday with Gallups, and we have our own little segment on there. And it really is an amazing thing to watch how you guys have come on board with us and supported us, and we are so grateful that you guys have decided to partner with us and to watch our material and grow in the knowledge and strength and power of the truth of the gospel of Jesus Christ. Because essentially that's, in the end, that's all that matters, right? That we're a family and we're moving forward in the kingdom work and doing the exploits that the Bible promises we would do, especially in the times we live in. So these little broadcasts we're going to cover things in the news with respect to prophecy and science and all of that. So welcome, and we're glad you're on board with us. So let's kick the first one off with something that we've been known for a long time. And that is the news out of the science community, and there's great news even today. And so this is how this is going to work when I do it. We may have other faces come on board and do the show, but when I do it, it will probably involve going to the blog and picking a story that we know you guys will be interested in, and then just covering it and talking about it. And sometimes we may even get special guests, and they will appear through Skype, and they'll appear, you'll see them here on the screen, and I have the computer right in front of me here that I'm using, but they will appear on the big screen here, and we'll just talk and have a conversation, and we may get some people to join us over Skype and just talk and talk things out. So we look forward to doing that. But let's kick this one off with a story from the Institute for Creation Research. Now we love the Institute for Creation Research. They can be found at icr.org. And listen, they are on the front lines of doing the battle and pushing back the frontiers of scientific ignorance. And this whole evolution thing, you know, even Richard Dawkins admits that evolution is the engine which drives modern atheism. So, you know, we all have certain assignments from God, and our assignment is to push that back, and to do exploits that come against the child abuse that is so prevalent today. Teaching children they are animals is child abuse. And if we don't stand up for the children, who will? Who will? We have the truth. We know that people are not animals. People were distinctly created apart from the animals in the image and likeness of God. And so we know the science on this, and we're digging it out and giving it to you. So here's the story out of the Institute for Creation Research today, in fact. And essentially the headline is Bacterial Evolution is Actually Design in Action. And we've talked about this many times, and it's a giant embarrassment for evolutionists. The evolutionary community has been buzzing over bacteria's new ability to obtain citrate, a carbon-containing chemical, from their environment and use it as a food source. Some say this confirms evolution in action, but what if the bacteria were designed to modify themselves? That might disappoint evolution enthusiasts, wouldn't it? Well, in fact, it does disappoint them, and it's quite an embarrassment for them. So let's just read on here. Microbiologist Richard Lenski is renowned for managing the most extensive and intensive evolutionary experiment on bacteria. Over several decades, his team has tracked changes in over 40,000 generations of E. coli Although these bacteria normally can't import citrate in the presence of oxygen, after 30,000 generations, some of his did. Did the bacteria invent a new mechanism to import citrate? And if so, then how? And if it did, why does that show evidence for evolution? If things are doing just as they're designed to do, how does that show evidence for evolution? Because evolution is the presupposition for these people. Let's read on here. To verify that this new trait represents the kind of evolution that can change a microbe into a man, researchers needed to describe exactly what went on behind the scenes. The highly anticipated details appear in the journal Nature. Okay, but prior to this new trait, none of the other observed changes that Lenski and his team had tracked down showed that bacteria evolved into a different basic kind. They were still E. coli bacteria, or even bacteria itself. And so far, none of those changes specified any new functional coding, which would occur the same as if chance-based phenomena could. Write a computer program, for example. Did the strain of E. coli that acquired the new ability to import citrate, called CIT+, construct new functional biochemical machinery by chance-based mutations? Well, let's find out. In 2010, biochemist Michael Behe reviewed 12 new phenotypes, which are outward expressions of genetic coding that Lenski's E. coli displayed from 1994 to 2008. Behe categorized that the known genetics producing each new bacterial phenotypes as either losing, shuffling, or gaining what he called functional code elements, which include genes and gene promoters. All the known changes in the bacteria were either a loss or reorganization of pre-existing functional coded elements. None of the new phenotypes came from a gain of functional coded elements, and yet this is what molecules to man evolution requires. Let me go through that again. In order for microbe to man evolution that goes beyond the genus, beyond the kind, beyond the barrier that keeps these creatures as bacteria, they would have to find functional coded elements and all known changes, and so producing each new bacterial phenotypes as either losing, shuffling, or gaining what he called functional coded elements. Okay, that's what's required in order for microbe to man evolution to even be possible. But all of the known changes in the bacteria were either a loss or reorganization of pre-existing functional coded elements. None of the new phenotypes came from a gain of functional coded elements. Yet this is what molecules to man evolution requires. Well, at the time, the mechanism underlying the citrate-eating phenotype was unknown. Behe wrote, if the CIT+ phenotype is due to one or more mutations that result in, for example, the addition of a novel genetic regulatory element, gene duplication with sequence divergence, or the gain of a new binding site, then it will be a noteworthy gain of FCT, that's functional code element mutation. So the big question here is, did E. coli evolve into a CIT+ strain by natural selection? Or did mutations construct new and functional coded elements to its DNA? If so, it would be the first in recorded biological history. If not, then it would just be another loss or modification of a pre-existing piece. In Lenski's experiment, the bacteria, both CIT+ and wild type, already possessed a gene named CIT-T. It encodes a protein that it transports a range of citrate-like chemicals. The recent results show that the bacteria made extra copies of CIT-T and neighboring sequence, a process called gene amplification. More copies of the gene should translate to higher amounts of the transporter protein that it encodes. With enough transporters, the bacteria could access enough citrate. But oxygen deactivates the CIT-T, and having many copies of a gene that is turned off is not very useful. But the bacteria solved this problem. The bacteria solved the problem when the amplification event also moved the gene sequence to a different place in the bacterial chromosome where a different but pre-existing promoter could regulate it. Unlike the original one, it appears that the new promoter does not have an oxygen off switching mode. Instead, it allowed expression of CYP in the presence of oxygen, and this was the big deal here, so that the bacteria successfully imported enough citrate to grow. The study authors wrote that the structure of the CYP amplification led us to propose that the CYP plus CYP trait arose from an amplification-mediated promoter capture. Further investigation confirmed the proposal. So, the bacteria solved the problem of accessing the alternative food source, which was citrate, by generating extra copies of the citrical gene and by placing those copies under the control of an appropriate promoter. Does any of this resemble natural, undirected Darwinian evolution? Really? I mean, it doesn't really. I mean, this amazing mechanism invented no new functional coded elements. It merely modified pre-existing elements. Therefore, not only did the CYP plus bacteria not evolve into molecules to man directions, but they showed that what could only be ingenious DNA rearrangement mechanisms. And what mainstream headlines portrayed as evidence for evolution is actually the opposite. It's evidence for intelligent design. And intelligent design requires an intelligent designer. So thanks to the Institute for Creation Research for putting this into proper perspective, and we'll put a link to the article and all of the scientific references in the video description here. But hey, listen, you don't have to buy that microbe to man evolution, deep time evolution. You don't have to buy any of that, either theologically, philosophically, or even scientifically. You don't have to buy it. And the mechanism of God did it is always the best way to explain why we're here, why biological systems act the way they do. And I mean, thank you to the Institute for Creation Research for bringing this out to us. And thanks everyone for watching the very first broadcast of PPSIMMONS Television. And we look forward to bringing you more great stuff moving forward. Thanks for watching.